Klebsiella pneumonia is an opportunistic gram-negative bacterium that has become a serious threat worldwide. Unfortunately, there is no consensus for an ideal reference gene for RT-qPCR data normalization on K pneumonia. In this study, the expression profile of 11 candidate reference genes was assessed in the cells submitted to various experimental conditions. But before we go any further, let me introduce myself and my partner. Hi, I'm Lorna, and this is my partner Moose. Here in this video, we're going to talk about the issues related to the topics, mechanisms, as well as application. So the problem that we're having here is it becomes a global threat due to the emergence and spread of hypervirulence. However, there is no ideal reference gene that meets the mentioned criteria, which prompted them to perform a systematic approach to identify the reference gene to be used on RT-qPCR analysis. There are four mechanisms that we're going to talk in this video, which are how reference gene is selected, how they assess the stability of the gene, as well as identification and expression of variability genes. So in the selection of the candidate reference genes, in the first mechanism of this study, 11 reference genes were selected and evaluated as potential candidate reference genes for RT-qPCR analysis in K pneumonia. This table shows the primer sequences specific for each selected candidate gene and the corresponding size for the amplicon for K pneumonia strain ATCC10031. This table also displays that the PCR efficiencies range from 83.4% for pyrroline 5-carboxylate reductase to 98.2%. Overall, the selected genes presented minimum cycle variation across all samples, as indicated by the low standard deviation of the mean CT. Most genes showed cycle variation below 1 cycle and none above 2 cycles. Overall, the selected genes presented minimum cycle variation across all samples, as indicated by the low standard deviation of the mean CT. Most genes showed cycle variation below 1 cycle and none above 2 cycles. The expression stability of the 11 genes placed in different conditions and analyzed using different statistical algorithms such as BestKeeper, GNorm, NormFinder, DeltaCT, and RefFinder. Two genes which are phenylalanine tRNA protein transferase and tryptophanyl tRNA synthetase 2 were excluded after preliminary analysis by BestKeeper and GNorm. According to BestKeeper, genes with standard deviation values greater than 1 are considered inappropriate as reference genes. The remaining 9 genes were submitted to the Pearson correlation coefficient calculation by BestKeeper and to GNorm, NormFinder and RefFinder programs. This table summarizes the statistical calculation generated by BestKeeper, GNorm, NormFinder, DeltaCT, and RefFinder programs and shows the ranking order of the genes with the most stable expression across all different conditions tested. As shown in the table, BestKeeper indicated BRCA, Rho, and glutamine synthetase A are most stable. Genome analysis indicates that all tested genes have stable expression across samples. As shown in the table, NormFinder indicated recombinase A, Rho, and Pro-C as the top-ranked genes with stability values. The recommended comprehensive ranking by RefFinder indicates BRCA, RPOD, and Rho as the most suitable genes. Moreover, DeltaCT analysis performed by RefFinders show BRCA, Rho, and Pro-C as the top-ranked genes. Bioinformatics analyses were carried out to identify putative full binding sites on the chromatin region of genes related to iron acquisition system in K pneumonia. This table shows the putative fur boxes identified on the upstream region of the genes SIR-A, RR, and FRU, which encode the caryophyllate-type cytophore receptors SIR-A, FET-A, and FI. Figure A shows that all putative fur boxes identified on this study rendered red A. E. coli H1717 colonies on the concave plates, which confirms that E. coli fur repressor was able to bind the cloned putative fur boxes in an iron-dependent manner. As shown on Figure B, the pneumonia purified His fur protein was able to interact with the DNA fragments containing the putative fur boxes in the presence of divalent manganese ions, which resulted on a mobility shift of the DNA fragments. Addition of EPT abolished the mobility shift of the fragments, indicating that divalent cations are required for fur interaction with the promoter region.
And this is the last mechanism where they utilize the top two most stable reference gene REC-A and Rho, as well as FFH gene, which is the least stable gene, and with RRSH gene, which is commonly used as endogenous control.
These are the promoter regions found in cane pneumonia.
This figure shows when they utilize REC-A and Rho, it reveals upregulation of 3 promoter in iron-limiting condition and downregulation in iron-replete condition when compared to the control condition. Similar results were obtained when the data were normalized with REC-A and Rho and with the REC-A plus Rho combined as a normalization factor. On the other hand, normalization with FFH and RRSH yielded inconsistent results. When FFH gene was used, CRI appears upregulated at both iron-replete and iron-limiting conditions. When RRSH was used as normalizer, no significant difference in the expression pattern of 3 promoters was observed at iron-limiting conditions.
RT-qPCR is used for nucleic acid quantification. So, for example, in this study, it was used to employ access to the expression of promoter regions in cane pneumonia cells subjected to iron-rich and low iron conditions. It is used to find the best reference genes. So, in this study, they found 4 out of 11, which are REC-A, Rho, RPOD, and PRO-C as reference genes, with Rho and REC-A being the best ones for gene expression normalization in cane pneumonia. The normalization of RT-qPCR data using either REC-A and Rho individually or as a combined normalization factor resulted in similar and reliable results. Used as normalizer of the relative expression of genes related to iron uptake systems in cane pneumonia cells submitted to iron-replete and iron-limited conditions. Since iron uptake is controlled by fur protein and iron availability is crucial for both survival and virulence of pathogenic bacteria, this finding can help in reduction of pathogenicity in cane pneumonia. The purpose of this study was to find the best gene for bacterial gene expression using qPCR, which is the standard method used to analyze gene expression due to its specificity and sensitivity. Although one error observed is failure to confirm the constitutive expression of the references genes used to measure the relative expression of genes of interest. So the finding of this reference gene can be used to study the regulation of iron uptake in cane pneumonia, which can help to reduce its pathogenicity.
Hi, this is Lorna.
And this is Mush.
Thank you for watching our video, sir. Hope you enjoyed it.